Good morning, everyone. At the outset, I would like to thank acadeMe for inviting me over here in my own country with my own colleagues on a beautiful Sunday morning to speak on a topic which is real-world evidence, a focus on gonadotropins. This is my disclosure. Now, what is the role of gonadotropins? We know that urinary and recombinant products are equally recommended and routinely used even by ASHRAE guidelines. But definitely there is differ with respect to the origin, production, purification methods, and this in turn may vary with the purity, variability of isoforms, glycosylation profile, sialic acid residue compositions, and isoelectric coefficients. These variations may then affect the bioactivity, metabolic clearance, and half-life, which is very important, and this can potentially lead to differences in the therapeutic effectiveness in women undergoing ART treatment. We as clinicians have many choices of gonadotropins and which to select. Definitely the patient profile comes first and the cost effectiveness. In a country like India, where 95% of the patients are paying from their pockets, not funded by the government or the insurance company, the cost becomes a major concern when the patient comes for IVF. Efficacy, very important, and evidence from RCTs, what we are listening and what we follow. Ultimately, it's the patient's choice. Now, let us see how we can combine an RCT and a real-world evidence. The RCTs have a high level of internal validity, but these are on ideal conditions. I myself have been a part of a number of RCTs where there are strict inclusion and exclusion criteria where PCOS, endometriosis, ovarian cystectomy patients, age more than 37 are all excluded. And the ideal patients are studied when we are comparing one drug with the other. But the challenges are immense because we have variations in patients' characteristics and treatment patterns. So RCTs may not fully represent the complexities and the variations observed in the real-world patients' evidence. So what is real-world evidence? There is collection which includes efficacy and safety data in real-world setting. We can get a patient of 42 years with severe endometriosis, chocolate cyst, which were not included those in the RCTs. So it is very valuable to have this evidence for healthcare decision making. It also provides insight into drug and medical product efficacy, safety, usage patterns, and definitely it overcomes, most importantly, the limitations of RCTs, providing valuable solution to their drawbacks. So today we can see and we can understand how RCTs and real-world evidence complement each other to improving the care given to our patients. If we look at the RCTs that are existing right now about exogenous FSH, they have mentioned that there is no significant difference in the pregnancy rate per cycle started between recombinant FSH versus HMG or versus FSH purified or versus FSH highly purified. No potential difference is noted, whereas others have noted that there is a potential difference between the two when you compare Rec FSH with HMG. Again, there is one more study which shows that compared to the originator, biosimilar Rec FSH alpha preparations have shown no clinically meaningful differences in efficacy, and the outcome measured was the number of retrieved oocytes. And finally, there is one more from Chua et al., which is very recent in 2021. It's a recent systemic review and meta-analysis of randomized controlled trials which demonstrated lower probability of live birth and pregnancy, both clinical and ongoing in couples treated with Rec FSH biosimilars compared with the Rec FSH alpha originator. So when you are deciding a drug and we have confusing RCTs, what do we do next? And we all know that in our clinical practice also, we don't follow blindly, as Sash said, the RCTs. We have to understand which is good, which is bad, and apply it and then only confirm it. So let us look at the real world evidence. Before that, I'll compare the originator versus the biosimilar. Whenever an originator comes into market, it needs to establish the quality characteristics, biological activity, safety, and efficacy based on comprehensive comparability study before it can be approved for use. So biosimilars with reference to REC-FSH alpha have been approved by the European Commission based on the respective comparative studies which I recently just mentioned, showing similar efficacy in terms of number of oocytes retrieved. But further head-to-head clinical trials as real-world evidence studies are needed to assess the comparative effectiveness, clinical effectiveness, and the potential differences in the reproductive outcomes between the reference REC-FSH alpha and the biosimilars. So this one is a huge observational study from the French nationwide database, which has comparative effectiveness of gonadotropins used for ovarian stimulation during ART in France. This was to describe the ART pathway in France, compare the live birth rate, cumulative live birth rate of the gonadotropin products used for ovarian stimulation in France, and also to report the serious adverse events leading to hospitalization. So from 2013 to 2018, the data was collected. Until 2019, it was followed. And there was a huge number of patients, 153,000 women undergoing 245,000 cycles. Almost 99% of the population was covered. And the data was robust because it had evidence and it was individualized from the billing and reimbursement of the outpatient healthcare and from the National Hospital Discharge Database. Out of these patients, there were 45% with REC-FSH alpha originator, 20 was the beta originator, 30% was urinaries, mainly HP-HMG and urinary FSH-HP, and 5% was with biosimilars. And when they compared the REC-FSH alpha originator versus the urinaries, they found that the urinary product, the patients when it was used, they had a 7% lower chance of live birth weight compared to the originator, REC-FSH alpha, and the cumulative live birth rate was even lower to 11%. And when it was compared, the originator versus the biosimilar REC-FSH alpha, the live birth rate was 19% lower in the biosimilar when it was compared to the originator, and the cumulative live birth rate was also lower by 14%. So this is the first nationwide French study to compare live birth and cumulative live birth rate between the outcomes of different gonadotropin products, which showed that the urinaries and the biosimilars had a lower probability of live birth rate and cumulative live birth rate compared to the REC-FSH alpha originator. This is the study, a non-interventional study in Germany, where the comparative effectiveness of REC-FSH alpha versus highly purified urinary human menopausal gonadotropin, HMG-HP, in ART cycles. So this was data from 71 German IVF centers. The women had all undergoing the first stimulation cycle, and where the ovarian stimulation was performed with REC-FSH alpha or HMG between 2007 and 2010. A huge data again with 28,000 first cycle women undergoing it, out of which 61.9% was with the REC-FSH alpha and 38% with the HMG-HP. And the primary outcomes that they analyzed were cumulative live birth rate, cumulative ongoing pregnancy rate, and clinical pregnancy rate. And what they found, you can see there that it was almost all this, the per complete cycle live birth rate was higher in the REC-FSH alpha compared to the HMG-HP. And if you see the cycle cancellation rate, it was higher with the HMG-HP as it was compared to the REC-FSH alpha. So this study again concluded that when you are using the REC-FSH alpha, there was increased cumulative live birth rate, clinical pregnancy and ongoing pregnancy rate, and most importantly, there was a lower cancellation rates when it was used. And cycles with REC-FSH alpha ultimately had a shorter time to live birth, while time to live birth measured in weeks was similar between the two treatment, especially when it was in the GnRH agonist. So when even the agonist cycle, which was more prevalent at that time, was analyzed, it was consistently found that the REC-FSH alpha had a higher ongoing and lower cancellation rate compared to the HMG-HP. And finally, this is a recent retrospective matched case control study of recombinant LH versus HMG supplementation on FSH during controlled ovarian stimulation in the GnRH antagonist protocol. This was in the Taiwanese women. It was a retrospective single center study where women who received antagonist protocol and had REC-FSH with REC-LH for more than 5 days or REC-FSH plus HMG for more than 5 days, and the data collected from 2013 to 2018. So out of the 503 cycles, 256 were with antagonist protocol, and out of this, 166 received REC-FSH plus HMG or REC-FSH plus REC-LH for more than 5 days. And the outcomes measured were estradiol level, endometrial thickness, oocyte retrieved, mature oocyte, fertilized oocytes, number of embryos, grade of the embryos, implantation, pregnancy, live birth rates, miscarriage rate, and rate of ovarian hyperstimulation. And the results supported that the hypothesis that the treatment of REC-LH and REC-FSH can improve controlled ovarian stimulation in the IVF and ICSI cycle. So then comes our own study in India, where we did a retrospective analysis of the clinical outcomes in patients receiving originator folitropin alpha and folitropin alpha biosimilars in real-world clinical practice. So when it comes to our country, it is first the finances that we see, and many of our cycles starts with just HMG. The REC-FSH, whether it's the originator or the biosimilar, becomes a smaller group. And when we did this analysis of patients who had undergone IVF between 2016 to 2020, and they had been using the REC-FSH, either the originator or the biosimilars, we found that there were 364 such patients, with 174 treated with originator folitropin alpha and 190 with folitropin alpha biosimilars. And the main criteria in selecting the gonadotropins, as I said, was the affordability and advanced age of the patient, with of course patients coming with failure cycles and different drugs used in the previous cycle. We found out, and this was an analysis done by my research people, and they analyzed and they came to me saying that, Madam, the number of gonadotropin used is slightly higher in the folitropin alpha, but the results are much better than compared to the biosimilars. And it was an eye-opener for me when the final analysis was done. The average amount of gonadotropin used was 3000 IU in the originator group versus 2700 in the biosimilar group, and the dosage decided was based on the age, AFC, AMH, BMI. On day 1 and day 2 of the IVF cycle, we even measured FSH. The ovarian response was typically followed by follicular tracking with ultrasound scans, and the primary outcome, we did not stop at the oocyte retrieval and the quality of oocytes, but we measured the clinical pregnancy rate and the live birth rate. Of course, we did measure the oocytes retrieved, the number of embryos both on day 3 and blastocyst, proportion of women with good quality embryos in both the groups, number of embryos transferred, and we also measured the miscarriage rate. And we were obsessed with using in advanced age, so further we sub-analyzed it with patients under the age of 35, what was their outcome, and also above the age of 35 years. So you can see in the first graph over here, where we had the folitropin alpha giving us 4.3 embryos per patient compared to 2.5 in the biosimilar group. And 3.5, sorry, and the number of good quality embryos in the percentage of patients we see, the polytropin originator group had 82% patients with good quality cleavage stage and blastocyst, whereas the biosimilar group showed 68% patients only having good quality cleavage and blastocyst embryos. We compared then the pregnancy outcome overall in less than 35 and more than 35 years. In more than 35 years, it is the second column there, almost 31%, 46% had a clinical pregnancy outcome, which was up to 45 years, and 14% only in the biosimilar. So the difference is huge when we compare the pregnancy outcome in the advanced age women, but also in less than 35 years age group, we had 59% who had clinical pregnancy rate compared to 39% with the biosimilars. So here also REC-FSH originator outperformed the biosimilars when the pregnancy outcome was compared. And finally, when we analyzed the full-term live birth, in less than 35 years of age group, there were 43% live birth rate in the originator, vis-a-vis in the biosimilar, it was 17% only. And we compared more than 35 years age group patient, there was 31% live birth rate compared to 9.3% in the biosimilar group. And this was a very, very huge I would say information to us based on the real world evidence. Because in younger age group, we feel that the patients are young, the AFC, AMH is good, so any gonadotropin will work, because we have heard and we have read RCTs that all gonadotropins are the same. But when we analyzed in our own settings, it was totally blinded, only the clinical research persons did the analysis, and to our surprise we found that yes, the originator had a much higher chance compared to the biosimilar. And when we looked at the number of oocytes, we found that the majority of patients in both cohort had 11 to 15 follicles on trigger day, originator 39%, biosimilar 41, almost the same. The originator cohort had a number of patients with less than 10 follicles compared to the biosimilar. The patients had more cohort with 15, 20 or 20, more than 20 follicles. And despite having fewer follicles, patients receiving the originator had better quality embryos than patients receiving the biosimilar. So these findings indicate better reproductive outcomes with polytropin alpha originator in comparison to its biosimilars. And not only in advanced age, even in the younger age group, this has a better outcome as far as clinical pregnancy rate, live birth rate is concerned. And even the miscarriage rate you can see was almost the double in younger patients when biosimilar was used, that is almost 19% compared to 10% in the originator. So there is now a growing agreement that the connection between ongoing pregnancy and live birth rate is very important. And when you are analyzing the ART outcomes, it is the live birth rate that measures, and now even healthy live birth rate that measures, and comparing just the number of oocytes or the quality of oocytes is not the answer. So this result showed that patients receiving originator polytropin alpha had better pregnancy rates, live birth rates than the biosimilars in a real world setting. And real world studies comparing both FSH preparation should be carried out, not just the RCTs, to aid the clinicians and patients while choosing which polytropin alpha to use in ART. Overall conclusions, I will say that biosimilars and urinary gonadotropins, as far as the French study goes, has demonstrated a lower live birth compared to originator REC-FSH alpha and beta. When we look at the Taiwanese studies, we find that the REC-FSH, when it is supplemented with REC-LH rather than HMG, it had improved oocyte retrieval, mature oocyte fertilization rates while maintaining comparable implantation, pregnancy and live birth rates. And the German study from Buller et al. Shows that cycle stimulated with REC-FSH alpha showed higher live birth, lower cancellation rates compared to HMG-HP in all settings, including the GnRH agonist treated population. And finally, our own study concluded that REC-FSH originator demonstrated higher pregnancy rates higher live birth rates compared to the biosimilars in real-world evidence. So these results from the real world are contradictory to results from some RCTs, highlighting the importance that RCTs are definitely a gold standard, but there is some importance of using the real-world evidence to complement these RCTs in order to see the full picture and make it beneficial to our patients. Thank you so much.